and get into a little bit of the brain and kind of talk about gray matter and white matter. Now, we know what gray matter is and we know what white matter is, but just a little bit of a review here, because as we move through the central nervous system, all right, we are going to, it's important that you know where we're going to find, all right, gray and white matter. And so when we're talking about the central nervous system, we're discussing the brain and spinal cord, obviously, and we're going to talk about now where we will find the gray and the white matter for both of these structures. Well, since chapter 13 is on the brain, we'll talk about the brain. Then when we get to the spinal cord, we'll talk about where the gray and white matter is located in the spinal cord, chapter 14. So quick review, gray matter is going to be the cell bodies of those neurons, and that includes any and all processes coming off of those cell bodies, which are going to be the dendrites here. And then it is going to be made up of unmyelinated axons, because it's actually the myelin that gives, all right, that tissue the white color when we're talking about white matter. So when we discuss terms like the cortex or the nucleus, like the cerebral cortex, for example, this is that outer layer that covers the cerebrum. And that is made up of gray matter. So when folks are mentioning, all right, they're talking about the cortex, they're referring to the outer layer, the superficial layer of gray matter that you'll see on the cerebrum there. And that is going to be unmyelinated axons, cell bodies of the neurons, and dendrites there. And then a nucleus, like I said before, is a clustering of cell bodies. And we'll see, all right, when we're going through different parts of the brain there, we'll see you'll have this white matter, and then you'll see these little pockets of gray matter throughout. And that gray matter is going to be where these cell bodies are going to be located, and then we're going to see where one neuron will synapse on another neuron. White matter is a little bit easier to remember because that's just myelinated axons. That's it, nothing else. And so we call these myelinated axons in our central nervous system tracts. And I'll remind you again, in the peripheral nervous system, we call myelinated axons nerves. So here you can see a wonderful picture of gray and white matter. Right, here's the gray matter. It's on the superficial outside area of our brain here. So that's going to be made up of the neuronal cell bodies, right, unmyelinated axons, right, and our dendrites here. Also sprinkle in some glial cells. And you can see internal to that, we have our white matter. Here's a nice corpus callosum that's crossing from one cerebral hemisphere to the next. And then you have some projection tracts coming down this way. It's called our internal capsule. And then you'll see these globs of gray matter throughout. Those are the cerebral nuclei. Cool stuff. When we get into the cerebellum, we haven't quite touched on that yet, but keep in mind, similar type of situation. Our gray matter here on the outside, on the cerebellar cortex, and then our white matter on the inside. And then you can see here in our brainstem, throughout certain areas, same kind of situation. We'll see areas of white matter, and then you'll see pockets of gray matter on the inside there. And as we get down into the lower portions of the brainstem here, specifically the medulla oblongata, you'll see the same thing. We got some white matter on the outside, pockets of gray matter here. And then when we get to the spinal cord, you can see the spinal cord has the white matter on the outside and the gray matter on the inside. It's almost kind of like a flip-flop. When we're up in the cerebrum, it's the gray matters on the outside and the white matters on the inside. When we get to the spinal cord, we flipped the script, put the gray matter on the inside and the white matter on the outside. And you'll see why when we get into the spinal cord and we're talking about all the different tracts there. Okay, so we're going to kind of move away from the actual brain itself, but I want to talk about some of the association structures with our central nervous system. And that requires me to talk about what we call the cranial meninges. And the cranial meninges are these connective tissue layers, and there's actually 3 layers. And so they cover the brain and the spinal cord. And so one, they help to support those structures, but also they're going to separate, all right, certain elements in the central nervous system. And some of those elements will be like cerebral spinal fluid, blood vessels. And so within these layers here, we'll talk about this here in a few moments, right? We're going to see a separation of these tissues, and in some of these spaces that these layers will create, we're going to see blood vessels, cerebral spinal fluid. You'll see some other structures in there. So we're going to kind of run through all that here. So the cranial meninges are 3 layers of connective tissue. And we will start with the deepest, which is the Pia mater, the middle layer, which is called the Arachnoid mater, and then the outer layer is the Dura Mater. Mater, this is misspelled. There should only be 1 T here. Mater stands for mother. So Pia is soft, so that stands for soft mother. Arachnoid is spidery mother, and then Dura is hard mother. That's my favorite. The Dura Mater. Okay, that's the hard mother. So the innermost layer is made up of a loose connective tissue. And so if you recall, when we were learning about the different connective tissue coverings of a nerve and a muscle, right? Again, similar pattern. We saw 3 layers, and the innermost layer, right, when we were talking about those connective tissue coverings, is usually made up of loose connective tissue, that areolar connective tissue, and that's what the Pia mater is. It's a thin layer of the areolar connective tissue. The next layer, as we move from the innermost out more towards the superficial, is the arachnoid mater. So this lays right on top of the Pia mater, and there's 2 things worth mentioning here. We have a structure called the Arachnoid Trabeculae, and it's basically this Arachnoid Trabeculae are these extensions that extend from the arachnoid mater to the Pia mater. And it makes almost like, it looks like almost like a spider web. I'll show you a picture here in a moment. And so we call that the trabeculae. And in that space between the Pia mater and the arachnoid mater, we give that a name. We call that the subarachnoid space. And this is very important. You need to know this. This is where the cerebral spinal fluid is located. So have you ever heard of the term a spinal tap? That's when they go in and they take a sample of the cerebral spinal fluid. They got to get a needle into this space, into the subarachnoid space. And so I'll show you this in a moment here. All right. So the arachnoid mater, the spidery mother, we call it that because we have all of these collagen and elastic fibers that make up this layer. It looks like a web. Now, on the other side of the arachnoid mater, as we move towards the Dura Mater, we have another space. Now, this is a potential space. It's called the subdural space. Potential space meaning it should not be in existence unless some sort of trauma has occurred. So if you take a blow to the back, let's just say, well, it doesn't have to be the back, since we're talking about the cranial meninges, right? Again, scenario, you're riding your bike without a helmet, you fall off the helmet, you fall off the bike, you hit your head. And you damage one of the blood vessels, right? That's here in the cranial meninges, and that blood vessel bleeds into the subdural space. It will fill up with blood if that blood vessel is damaged. So it's a potential space, all right, until some sort of trauma occurs, and then it actually becomes an actual space because it's filling up with blood. All right, the other last type of meninge is the Dura Mater. The Dura Mater is made up of dense irregular connective tissue. And this dense irregular connective tissue has 2 layers. Now, a lot of the Dura Mater, both of these layers are fused together. But in some scenarios, all right, they separate. And we'll talk about what happens when they separate. So the first layer, the layer that is deeper, is the meningeal layer, and then the more superficial layer is called the periosteal layer because, yes, it forms the periosteum on the inside or internal surface of our cranial bones. So that periosteal layer, right, on the inside of those cranial bones is going to be part of our Dura Mater. Now, when the two layers of the Dura Mater separate in certain areas, they're purposely supposed to be separated, they form these structures called dural venous sinuses. And these sinuses will drain the blood from the brain. All right, so in these areas where the meningeal layer and the periosteal layer separate from one another, they become blood-filled spaces, the dural venous sinuses. And these dural venous sinuses will drain blood away from the brain. Then on the outside of the Dura Mater, we have another potential space. We call that the epidural space. And in this space, you will have arteries and veins. Similar type of scenario. If you have experienced some sort of trauma to your head, you have the possibility of damaging these arteries and veins and getting an epidermal, what we call an epidermal hematoma. And that's when these blood vessels are damaged and blood starts to fill up that potential space outside or superficial to the Dura Mater there. So if we zoom in here and look at our cranial meninges here, look at the different layers. Here's your skin, your scalp, right? And then you have your bones underneath, okay? So remember, our cranial bones are flat bones. Flat bones don't have a medullary cavity, so they're made up of compact bone and our spongy bone. It's our peanut butter sandwich here, okay? So here's our peanut butter, that's our spongy bone, and then the compact is our actual bread. Well, keep in mind, all right, bone has periosteum on the outside. So you have a periosteal layer on the outside of the bone, but then on the inner layer, or on the inside here, you have the periosteum on the inside of our cranial bone. Well, on the inside here, that periosteal layer becomes one of the two layers of our Dura Mater. So you can see here, here's our Dura Mater, it's fused together. And then our periosteal layer and our meningeal layer separate from one another and they create a space. And the space is one of our dural venous sinuses, and that's where, all right, blood will drain from the brain into this area. And then it will eventually drain out of your skull into your neck. So we go a layer deeper, we have our arachnoid mater right here. And then the space between our Dura Mater and our arachnoid mater is called the subdural space. It is a potential space. Now it looks like an actual space here because we just wanted to show it to you in our illustration. And then you can see you have all of these arachnoid trabeculae here that extend from the arachnoid mater to the pia mater. And the pia mater is this purplish colored layer that sits right on top of our nervous tissue here. So it's drawn, it looks like it's pink or purple to me. And so that's the pia mater that sits right on top of the brain. So this space here between the pia mater and the arachnoid mater, right, that space there is called the subarachnoid space. That's where your cerebral spinal fluid flows. All right, so we talked about the different layers of the meninges. And so now I would like to talk to you about some of these sheets, all right? When we're talking about the outer layer, the Dura Mater, normally it's fused together. Well, this tissue is made up of that dense irregular connective tissue. It's a tough tissue. And so we'll see throughout your cranial cavity, right, where this Dura Mater has formed these sheets, and it helps to divide certain parts of your brain from other parts of your brain. and it also provides a support role to our brain. So the first tissue is called the Falx Cerebri. And again, when we're talking about this tissue, this is what we call cranial dural septas. That's just the Dura Mater, the outer layer of the Dura Mater, of your cranial meninges, the outer layer there. So we'll see in the Falx Cerebri, the Falx Cerebri sits in the longitudinal fissure. In case you don't remember what the longitudinal fissure was, that's that space in between the right cerebral hemisphere and the left cerebral hemisphere. So you have this dense irregular connective tissue, this Dura Mater, that sits right there in that space. And we talked about how the Dura Mater is two fused layers of tissue. All right. Well, here at the Falx Cerebri, we'll see that those two layers separate a little bit to form some dural venous sinuses. So in the Falx Cerebri, we have two sinuses. We have what's called the superior sagittal sinus and the inferior sagittal sinus. So if I show you a picture here, right, here's a mid-sagittal cut side view. Okay, here's your nose, so this is anterior. And so we'll zoom in here. So here you can see how this is the Falx Cerebri. That's Dura Mater. See how thick that is? Oh, it's tough to see how thick it is, but you can see that it pretty much extends from the top of your skull, right, down by a few inches into the cranial cavity here. So if we're looking at it from the posterior view, here are the two the two hemispheres. Here's the right cerebral hemisphere, here's the left. And so in the center is the longitudinal fissure. And in that longitudinal fissure sits this thing, the Falx Cerebri. So at the very top, it separates, and at the very bottom, it also separates to create a space. And those spaces, the top one is called the superior sagittal sinus, and it drains blood from the brain up there. And then you've got the inferior sagittal sinus down here, and it drains blood from the brain down in this area here. I got more pictures to show you. Whoops, went too far. All right, so that's the first cranial dural septa. The next one is called the Tentorium Cerebellar, and I like it because it literally, like I noted here, it is a tent. And so it's basically a tent that sits on top of the cerebellum. Now, we haven't learned what the cerebellum is. That's that little mini brain that sits on the back inferior portion of your of the cerebrum there. And so the Tentorium Cerebellar is going to separate both the occipital and temporal lobes from the cerebellum. And so again, right, we're going to see some of these layers separate and create a sinus. So the Tentorium Cerebellar is going to create the transverse sinus, and that's going to go along the posterior border there of your skull. And we'll also see the straight sinus, which is in the mid-sagittal plane. I'll show you. That's going to extend kind of backwards from the inferior sagittal sinus. So taking a quick look here in our picture here. Here you can see, I'll zoom in. You can see a nice picture of the transverse sinus. So here's the cerebellum, and here is our cerebrum. And so basically the Tentorium Cerebellar sits, it lays right on top of the cerebellum, and it separates the cerebellum from the occipital lobe here of your cerebrum. And then it's tough to see, but on the side here is the temporal lobe. So it separates those two lobes from this region of your brain, from the cerebellum. And so the Tentorium Cerebellar separates those two layers there. And on the back side here, it forms the transverse sinus. And then you can also kind of see here, it's tough to see, it's not the best picture, the straight sinus. Here's the inferior sagittal sinus. If you follow it back, it comes back here, and then then you can see this one structure coming backwards here. That's the straight sinus. You can see part of the Tentorium Cerebellar here. Sitting, here's your cerebellum. And so it sits right on top of it like a tent flap. All right, two more. The Falx Cerebelli and then Diaphragma Cellae. All right, so the Falx Cerebelli is going to separate your left and right cerebellar hemispheres. We have the Falx, look at notice the spelling here. Cerebelli for cerebellum, and then cerebri for the cerebrum. All right, so the Falx Cerebri separates your right and left cerebral hemispheres. Then our Falx Cerebelli is going to separate your right and left cerebellar hemispheres. And so the Falx Cerebri will separate and form the occipital sinus on the backside here. Then one last cranial durosepta, and that's the Diaphragma Cellae. This is the smallest one, and this makes a nice little covering for our pituitary gland. Now, in lecture today, we learned about the pituitary gland's relation to the hypothalamus. Right, so that hypothalamus actually has a physical connection to the pituitary gland. And if you recall, the pituitary gland sits in the sella turcica of the sphenoid bone. Remember our Turkish saddle? So it sits in that little, if we're looking at it from the side, here is the sella turcica. And so the pituitary gland will sit in it like this. And then you have the infundibulum, also known as the pituitary stalk. It comes out and will physically connect the pituitary gland to the hypothalamus above it. Well, you have this little covering that sits right over here. And that's the Diaphragma Cellae. And then real quick, again, it's tough to see here on this drawing, but our Falx Cerebelli will divide your left cerebellar hemisphere from your right cerebellar hemisphere. And where it separates, it's going to form the occipital sinus. All right, so we learned about the cranial dural septae. We learned about the cranial meninges. So let's learn about another structure in the brain, and those are the brain ventricles. Ventricles, I've briefly mentioned the ventricles are spaces or cavities within the brain, and it will contain cerebral spinal fluid. And these ventricles are lined with ependymal cells. Back in chapter 12, we learned about the role of ependymal cells. The ependymal cells will generate and make cerebral spinal fluid. Cerebral spinal fluid is very similar to your blood plasma. It's not the same, but it's very similar. So all these ventricles are connected to one another. You've got the left and right lateral ventricle, the third ventricle, the fourth ventricle. And so all of these ventricles will connect to one another, and they'll also connect to your spinal cord's central canal. Well, what's that? Well, that central canal is going to be, you can't see it. It's really hard to see. I'll zoom in a little bit. Right there, see the little hole there? That's the center of our spinal cord. That is the central canal. It's a little hole. It runs all the way down the center of your spinal cord. And that hole, that canal is lined with ependymal cells. And that we will see cerebral spinal fluid there. We're also going to see cerebral spinal fluid in our subarachnoid space, that space in between the pia mater and the arachnoid mater. And we'll talk about some of the other places that we'll see the cerebral spinal fluid here in a moment. All right, so we have our four ventricles. Our left and right lateral ventricles, those are the large ones that we're going to see in the center of the cerebrum. And so they're separated from one another by a structure called the septum pellucidum. You'll need to know that. We'll do some labeling for lab class. But they are divided from each other, all right, with the structure called the septum pellucidum. Then both your left and right lateral ventricles will drain downwards inferiorly to the third ventricle. And that third ventricle I mentioned, we were going over the anatomy of the diencephalon, right? The third ventricle sits right in the center of the diencephalon. Essentially, the diencephalon will surround the third ventricle. So each lateral ventricle will drain into the third ventricle through a structure called the interventricular foramen, also known as the foramen of Monro, M-U-N-R-O though. Right. And then the third ventricle will drain down inferiorly into the fourth ventricle. The fourth ventricle is located in your brain stem, specifically behind the pons and in front of the cerebellum. And so the structure that drains the third ventricle into the fourth ventricle is the cerebral aqueduct. It's this long tube-like structure. Once we get to the fourth ventricle, here's the cerebral aqueduct, drains inferiorly. The fourth ventricle looks like a diamond almost. Down at the bottom here, it'll continue on into the spinal cord, and that will turn into the central canal. But we have these openings laterally on each side. And that is where our cerebral spinal fluid, some of it, will drain out or open up into the subarachnoid space. So cerebral spinal fluid will drain down into here, into the fourth ventricle. Then it's got a couple of options. Okay, it can go out to the sides there, or it can go inferiorly down into the spinal cord. We like options. All right, so here's a nice picture. Okay, a couple different views here. Let me zoom in. Here are the two lateral ventricles. This would be our right lateral ventricle. Here would be the left. Each one individually drains down into the third ventricle through the interventricular foramen right here. It'll drain down here into the third ventricle. And then the diencephalon surrounds the third ventricle. Then the third ventricle will drain down into the fourth ventricle. So at that point, cerebral spinal fluid can go out to the sides here, right? Or they can drain down further inferiorly down into our central canal of the spinal cord. So if you look, the lateral ventricles almost look like a oddly shaped C. See, it curves around, has this little structure shooting off the back, but also comes around to the front here. So it's kind of like a C shape. And if you look at it here from the anterior view, you can see, I always thought it looked like horns, right? Well, horns from a ram, right? So you can see the lateral ventricles drain into the third ventricle drains down into the fourth ventricle. Then the cerebral spinal fluid can go downwards or it can go out to the sides here. Well, you heard me talk about the ventricles and the fluid there. Let's talk a little bit about the cerebral spinal fluid. Like I said, it's very similar to our blood's plasma. Okay, so therefore, it is going to have certain consistencies similar to blood plasma, right? Except cerebral spinal fluid has more of a clear appearance to it. And so the two areas that you will see cerebral spinal fluid in normal circulation is going to be through the ventricular system and the subarachnoid space. Here's one of my favorite statistics or facts in this course. All right, cerebral spinal fluid has several functions. Helps with the brain's buoyancy, helps protect the brain, helps to create a stable environment for the brain when we're talking about the movement of nutrients and getting rid of waste. But one of my favorite stats is buoyancy. This, all right, the cerebral spinal fluid reduces the buoyancy of the brain by 95%. That is huge. There's several reasons why your brain doesn't just drain out of your foramen magnum, all right? I won't get into all of that. But this function of cerebral spinal fluid helps to keep your brain in that aquatic environment almost weightless, right? Because again, it needs a very, very, not a sensitive environment, but an environment that is going to not inhibit any of its functions. And so the cerebral spinal fluid is amazing in the fact that it helps reduce the buoyancy, but also it's putting your brain in an aquatic environment so it acts like a liquid cushion. In case you have to, you take some jarring maneuvers, like if you're on a roller coaster, for example, all right? That aquatic environment helps to protect the brain. Really kind of cool. So let's talk about a little bit about the formation of cerebral spinal fluid, because when we understand this formation, we're going to talk about some of the pathologies that can occur. So let's talk about how we make it. All right, back in chapter 12, we learned about the ependymal cells. The ependymal cells are grouped together, and these groupings that we see in these ventricles, we call a choroid plexus. And so it's this grouping of ependymal cells along with our capillaries, because essentially the ependymal cells are going to filter out the cerebral spinal fluid from your blood. So it filters out that cerebral spinal fluid and it modifies it. And like I said, it's very similar to our blood plasma, but we're going to see more electrolytes. For example, we're going to see more sodium, more chloride, but guess what? Less potassium, less calcium, all right? So interestingly enough, I like calcium. We learned about its importance, okay? And calcium levels need to be well regulated, but also calcium needs to be in certain locations, in certain tissues, and not in others. Because calcium has a tendency, if it gets involved in certain pathological processes, it can cause, for example, what we call dystrophic calcification. And you can get an accumulation of calcium into certain tissues, and it starts to create almost like a hardening of that tissue. And that could be very, very bad when we're dealing with neurological tissue here. So we'll see, again, like I said, cerebral spinal fluid is similar to blood plasma, okay, but it's not the same as blood plasma. All right, so we're going to see that generation of that cerebral spinal fluid, all right, in the ventricular system, and then it'll eventually make its way into the subarachnoid space. All right, so it's all in a big, huge circulation here. All right, so this cerebral spinal fluid, and for any of you folks that have ever had to experience a spinal tap, it happened to a friend of mine and a patient of mine just recently, in which they had undergone a spinal tap, and they, where they inserted the needle, it didn't heal up completely, and some of the cerebral spinal fluid leaked out. And unfortunately, because that cerebral spinal fluid leaked out, all right, it offset that equilibrium, that homeostasis there of that cerebral spinal fluid system. And the symptoms that people will complain of is severe. And I'm not talking about just a bad headache. I'm talking about severe headaches. That's how regulated the volume of the cerebral spinal fluid is. So your body has figured out a way to constantly resorb the cerebral spinal fluid because it has to be at a certain volume. And so it's going to circulate that cerebral spinal fluid. So what happens if your body, okay, if those ependymal cells make too much cerebral spinal fluid? We need to figure out a way to kind of open up a relief valve type of situation and get rid of that cerebral spinal fluid. So we have these structures that are located in your dural venous sinuses called arachnoid villi. Now they remind me of mushrooms. It almost looks like a mushroom cap. I'll show you a picture of it, but it's actually a couple of cells that overlap each other. So what will happen is when you've accumulated or produced too much cerebral spinal fluid, we're going to get rid of that cerebral spinal fluid, right, from the subarachnoid space, and we're going to offload it into the dural venous sinuses. Well, what's in the dural venous sinuses? Blood that you've drained from the brain. So this works out really nicely. You can see here, right, on this picture here, we have our live dissection. And here is our choroid plexus. And it's these structures that have, all right, capillary blood vessels and ependymal cells. And they'll start to produce that the CSF and pump it into the ventricles here. So if we zoom in, this is pretty much what it looks like. Here's your capillary. And then lining that capillary are the ependymal cells. And so they're going to allow certain things to escape from the capillary and into the ventricle cavity there. All right, so going back to what I was saying about the cerebral spinal fluid, if you take a quick peek here, we'll zoom in and show you what happens to cerebral spinal fluid. All right, so figure 1 here is showing you, right, we have our choroid plexus and it's developing and creating the cerebral spinal fluid and it's flowing through the lateral ventricles. Then it moves down here into the third ventricle. And that third ventricle will then drain into our fourth ventricle. Here's the cerebellum. This is the brainstem here. This is the middle portion of the brainstem, the pons. We'll talk about that, okay, in class later. So the cerebral spinal fluid will then drain down into the fourth ventricle, and it can continue to drain down into the central canal of the spinal cord. That's one area that it can drain into. Then it can drain either into the lateral aperture or the median aperture. Basically, those apertures open up into our subarachnoid space. Okay, so this space is all over throughout the cranial cavity. So the cerebral spinal fluid is going to travel all throughout that area, and it will come down here, right, into our spinal canal down in this area here. All right, but the point being is, all right, this subarachnoid space is all over the place. And so what will happen is, right, our cerebral spinal fluid will flow all throughout. And you can see these little areas. I can't zoom in anymore. I'll show you here in a second. But you can see these little spaces here called the arachnoid villi. And the arachnoid villi are these tissue structures that push into the dural venous space. And they're going to release that excess cerebral spinal fluid. So if we zoom in there, all right, here you can see, here's the subarachnoid space with our cerebral spinal fluid. So now we've increased the volume of our cerebral spinal fluid, so we have to release some of that cerebral spinal fluid. So the pressure builds up and it pushes open these cells here that just overlap one another. It creates like a one-way valve. And so as that pressure increases on this side, these cells will open up slightly, and that cerebral spinal fluid will then push itself out into, all right, our subdural space here, not subdural space, excuse me, our dural venous sinus space here. And then once that pressure decreases here, all right, these two cells will fall back down and close off, so nothing gets in, only stuff can leave. And that's how we get rid of that extra, right, cerebral spinal fluid. We just push it off into that region there. All right, so let's talk about, and we've already talked about the blood-brain barrier briefly, but let's actually, this is a good place to stop. I want to do some labeling today. All right, actually, I could go on and talk about this, but I want to stop right here. Yeah. Let's stop here with the blood-brain barrier, and we'll do some labeling here real fast. All right, so if you have your atlas with you, which you should, let's go ahead and pull that out, and we'll label what we covered in lecture today. All right, remember there's 4 regions to the brain. Just keep that in mind because it's really important, because on the test, if it says, what region of the brain is this pointing to, you need to say the cerebrum, because if it says what region of the brain is this pointing to, and you say that's the frontal lobe, yes, you are correct that that's the frontal lobe, but the question was asking the region. And there's 4 regions: the cerebrum, the cerebellum, the brainstem, and the diencephalon. So this arrow here is pointing to the cerebrum. The cerebrum is made up of 2 hemispheres, 2 halves. So we're looking at a posterior view of our brain here. This is the left cerebral hemisphere. Here is the right cerebral hemisphere. Right, you notice that there's a space in between. You can see it's nicely demarcated. All right, let me go back one. There, that space right here. We're going to talk about the longitudinal fissure here in a moment. So now we're going to break down the cerebrum into its lobes. There are 5 lobes to the cerebrum. So this arrow is pointing to the frontal lobe. Remember your boundaries of the frontal lobe. Here's the central sulcus. So the frontal lobe is all of this. Here's your lateral sulcus right here. So if the frontal lobe is all of this right here. Right? Down here below the lateral sulcus, this is the temporal lobe. This is where your ear is going to be. Again, another lobe of the brain. Here's the parietal lobe. The parietal lobe is posterior to the central sulcus. And then it sits right above the lateral fissure right here. Or I'm sorry, lateral fissure, lateral sulcus right here. So here is your parietal lobe, all of this right here. Then we have the occipital lobe right back here. And we're missing a lobe. Remember, there's a 5th lobe. It's the invisible lobe. So what we've done is, you don't have this picture, I apologize, it's not in the atlas, but when we pull apart, all right, the parietal lobe lifted up away from the temporal lobe, we can see inside, and there's the insula. That's for gustation, taste, yum, yum. Okay, so that blue line there that's highlighted with yellow, that represents the longitudinal fissure. And that is that deep depression that sits in between your left cerebral hemisphere and your right cerebral hemisphere. And what sits in that longitudinal fissure is that outer layer of the spinal, oh, no, excuse me, not the spinal, of the cranial meninges, the Dura Mater. That outer layer of the Dura Mater that sits in that space is called the Falx Cerebri. The Falx Cerebri occupies the longitudinal fissure. It partitions the hemispheres from one another, but it also helps to act to support the soft tissue of the brain there. Here's the transverse fissure. You can see the space a little bit better over here on the right. Okay, so that's that space in between the cerebrum and the cerebellum. That's the transverse fissure. Well, we have, right, the Dura Mater layer that sits in that area. That's called the Tentorium Cerebellar. The Tentorium Cerebellar. Going to the lateral view of our brain, right, here's the lateral sulcus. It's also referred to as the lateral fissure, so either or is acceptable. Which is confusing to me because a sulcus is usually not as deep as a fissure. There's the central sulcus. Now that arrow is not the arrow, excuse me. The arrow is pointing to that black line with the yellow highlight. That black line is occupying the central sulcus. I'm going to go to the previous slide so you can see the central sulcus is pretty, you can see it right here. That's the central sulcus. It's an important anatomical structure, all right, in the brain. So you see now the arrow is moved, and it's anterior to the central sulcus, and it's pointing to that gyrus there. That is your pre-central gyrus. The pre-central gyrus. That is your primary motor cortex. Voluntary movements all start there. Then behind our black line, behind our central sulcus, is our post-central gyrus. That's in the parietal lobe, and that is the somatosensory cortex. So conscious perception of senses winds up there. Post-central gyrus. There is the cerebellum, a little brain hanging off there at the bottom. That is another region to our to the brain there. And in between the two cerebral hemispheres is a structure called the vermis. That's right in here. The vermis sits in between the right cerebellar hemisphere and the left cerebellar hemisphere. This is a posterior view of the brain. I'm standing behind you in line looking at you. This is the view that I would see if I could see into your brain. There's the left cerebellar hemisphere. Then, if we mid-sagittal cut through the brain, you'll notice we have white matter on the inside of the cerebellum, and we call that arbor vitae. Tree of life, arbor vitae. Arbor, did I say arbor? Arbor. Right, that's the arbor vitae. And now comes the diencephalon. We'll finish up here with the diencephalon. And there's three parts to the diencephalon. Those arrows are pointing to all three parts. Just know, I'll go through each part individually here in a moment. The diencephalon is made up of the thalamus, the hypothalamus, and the epithalamus. All right, so first one, there's the thalamus. It's that oval-shaped structure. You have two. You have one on the right, one on the left. And this structure in the middle is called the interthalamic adhesion. This is what connects your two thalami to one another. Here's the thalamus on the other model. Right, then we have our hypothalamus. That's this area down here. The hypothalamus, also known as the master gland. Here you can see the hypothalamus on this model. And then the epithalamus, the last part, this arrow, remember the epithalamus is made up of the pineal gland and the habenular nuclei. Right here, it's pointing to the pineal gland. The pineal gland makes melatonin. Melatonin. There's the pineal gland on this model right here. And that helps to regulate your sleep-wake cycles. All right, I'm going to stop here because I know tests are coming up. People want to study.